Welcome to Manifested Publishers.
Welcome learners, my name is Stephen Kariungi and today we continue with our topic of discussion and the topic is genetics. We are looking at the last part of the topic and this is on practical applications of genetics, and the last time we identified 4 areas of application, and the first one was on plant and animal breeding, number 2 was in blood transfusion, number 3 genetic counseling, and number 4 crime detection. So today we are going to look at number 5, that is practical application number 5, and this is genetic engineering. Now, genetic engineering is whereby a certain desirable gene is first identified. After being identified, then it is altered or it's changed a little bit, and then it is isolated or it's separated from the organism where it is in existence, and then transferred into another organism of completely unrelated species. Another organism of a completely unrelated species. So we are saying that it is the identification of a desired gene, isolation, and transferring into an organism of an unrelated species. So this is the identification of a desired gene altering, isolation, and isolating and transferring the gene into another organism of unrelated species. So genetic engineering has been applied for quite some time in a number of fields. In the field of medicine, in coming up with vaccines, in farming to get rid of some pollutants, that is in the environment, also in farming to increase the productivity, and also in something that is called gene therapy that involves correcting defective genes. So you're saying that in farming, there is a certain hormone that is known as bovine somatotrophin that is used to increase the milk production. So the gene that is responsible for the formation of that hormone, bovine somatotrophin, is transferred into a genetically modified bacteria, and then that bacteria is able to produce this hormone in very large quantities for use to increase the milk production. So we are saying that a hormone called bovine somatotrophin which increases milk yield is produced by a genetically modified bacteria. in large quantities. For use in improving milk productivity. In medicine, genetic engineering has been used to make vaccines and hormones such as insulin, the insulin that is used to treat those who are suffering from diabetes. Of course, they inject themselves with insulin to try and raise the insulin hormone levels in their bodies, because possibly their pancreas is unable to produce the insulin hormone normally. So we have a number of areas, even in environment. There's a bacteria called Pseudomonas. Is used to break down hydrocarbons from petroleum, thus reducing pollution. So all those are cases of genetic engineering where a certain desired gene is identified, then it is altered, isolated, and then transferred into another organism so that it can either help to improve the yield, for example, in milk, in farming, in coming up with vaccines and hormones, let's say in medicine for treating various diseases, and also in environment to get rid of certain pollutants. The next area of application of genetic engineering is cloning. And this is the formation, this is the production of new individuals, new individual organisms without use of fertilization. And a very good example is Dolly, e.g. Dolly the lamb. So that Dolly the lamb was a sheep that was made without fertilization by use of cloning, whereby, for example, a cell was obtained from a certain part of the body, and then it was inserted into an ovum without a nucleus so that it can be implanted into another sheep's body without involving fertilization, and then it can give rise to an organism. So that is one case of cloning. Another application is in the human genome, and this is analyzing all the genes in all the chromosomes in the body, analyzing all the genes in all the chromosomes in the body to know, for example, the position of the gene that causes a particular disorder. For example, the gene that causes cystic fibrosis, the gene that causes maybe sickle cell anemia, where it occurs. And that one will help in, for example, what we call gene mapping or gene sequencing. Excuse me. This is important for gene mapping. This is just about identifying the gene location and gene sequencing, which is about identifying the sequence of nitrogen bases. So that marks the end of the topic of genetics, and we'll have an assignment based on what we have discussed. So the first question, what is A, gene mapping, B, gene sequencing, and number 2, explain the concept of genetic engineering and its application in farming and in medicine. So we are going to stop there, and until next time, goodbye.